Okay, the last lecture of the term. Finishing up chapter 16, the levels of protein. So I'm going to take you through primary, secondary, tertiary, and quaternary structures of a protein. So in the primary structure of a protein, it is the particular sequence of amino acids. Okay, in this particular example of the primary structure would be alanine bonded to leucine, bonded to cysteine, and then finishing off with methionine. So this would be considered the primary structure, the order of your amino acids. It's also called the backbone of the peptide chain, or the backbone of the protein. Insulin, this is a picture of insulin. It was the first molecule that we actually discovered the primary structure of. And it was determined to have two polypeptide chains that were held together by some cysteines. Now, cysteine is our only thiol amino acid, and if you remember, the only reaction you needed to learn about thiols was the reduction, where you lost two hydrogens, one from each of the thiols, and it made this disulfide bond. So here is two cysteines bonded together by a disulfide bond, and here's another cysteine bonded together by a disulfide bond. So anyway, so they figured out the primary structure of insulin. I don't expect you to know any of this, but it was pretty fun at the time. I remember when it was first sequenced. There are two secondary structures I want you to know. So I want you to know the alpha helix and the beta sheet. So a little bit about the alpha helix. The secondary structure of the alpha helix is a three-dimensional spatial arrangement of the amino acids in the peptide chain. So it's kind of the first way that the amino acids, once they're bonded together in that primary sequence, then they do they make different shapes with that big long chain. So one of the shapes they make is this alpha helix. The alpha helix is held by hydrogen bonds between the hydrogen of an NH group and the oxygen of a carbonyl group of the fourth amino acid down the chain. Now, I don't expect you to know it in that much detail, but that's what holds this alpha helix in this corkscrew shape. Is that here's a carbonyl group interacting with the hydrogen on an NH group on the backbone of the primary structure. Okay, so then the last bullet is a corkscrew shape that looks like the coiled telephone cord. So what do I want you to know about the alpha helix? I want you to know that it is a secondary structure. It makes that coil or corkscrew shape. And I want you to know that it's held together by hydrogen bonds. Okay, so you don't have to know that it's by every fourth amino acid, but I do want you to know that it's held together by hydrogen bonds, and that hydrogen bonds are occurring within groups off the backbone. Okay, pretty soon I'm going to get into the tertiary structure, and we're going to talk about the hydrogen bonding that happens with the R groups. But in your secondary structure, the hydrogen bonding is occurring between the backbone groups. Okay, so the second secondary structure I want you to know is the beta pleated sheet. Sometimes it's just abbreviated as the beta sheet. So the secondary structure of a beta pleated sheet consists of polypeptide chains arranged side by side. So what I'd like you to do, I'm going to go grab my pen, is it's one chain. So I don't think this picture does it justice, because what happens is the chain is going along and it's doing its thing, and then all of a sudden it does like this hairpin turn, and it comes back, and then it'll fold back on itself again and make another hairpin curve. So it kind of loops back and forth, and that's what your beta sheet's going to look like. There are hydrogen bonds also holding the chains together. So see, here's the oxygen on one carbonyl group and bonded to the hydrogen on an NH group. So this is where we get the hydrogen bonding. But once again, it's not happening between the R groups. It's happening between the backbone of that amino acid primary structure. So this side by side is kind of a big giveaway that they're talking about a beta sheet. The other one, the alpha helix, they often say the whole coiled shape is kind of a giveaway for that one. In a beta pleated sheet, the R groups are going to lie above or below the sheet. So the beta sheet, if you kind of think of it like this, your R groups are either sticking up or sticking down. It tends to be typical in fibrous proteins such as silk. Here's a picture kind of summarizing this. First one is the primary structure. So it is just the sequence of amino acids. The first amino acid, the second one, bonded together by what type of bond? A peptide bond, holding your amino acids together. And then, so that's the primary structure. These two are the two secondary structures that we're going to focus on. The alpha helix, which is the coiled, and the pleated sheet or the beta pleated sheet, which gives us the kind of the hairpin turns going back on each other. Once again, I do want you to know that here these dotted lines are representing the hydrogen bonding, and it is hydrogen bonding along the backbones of the amino acids. It's hydrogen bonds that hold those secondary structures in their particular shape. So here is a learning check. Indicate the type of protein structure, whether it's primary, a beta pleated sheet, or an alpha helix. So A says it's a polypeptide chain held together by side by side by H bonds. So that whole side by side, hopefully you said it was beta sheets. So A would be 3. B is the sequence of amino acids. So that's the definition of your primary structure. So the primary structure is the sequence of your amino acids. And here's the corkscrew shape, which goes with our alpha helix. That'd be number 2. Okay, moving on to tertiary structure. In the tertiary structure of a protein, it is considered the overall three-dimensional shape. So the way I like to visualize that is when I was growing up, it was back in the days that we had telephones that were actually hooked together by a telephone cord. Does any of you remember that? And we had, when I was growing up, we had a really long one. And the actual shape of the telephone cord, that corkscrew, would be considered our secondary structure, the alpha helix. But what would happen in my household is that cord was so long that every once in a while it would get tangled up on itself and make this big old blob of telephone, cord that you'd have to untangle at times. Well, when that cord like totally collapsed on itself into this kind of blob of a shape all knotted together, that would be an example of a tertiary structure. Okay, so they say it's the overall three-dimensional shape. Sometimes they say it's the way that the polypeptide folds on itself. It's another way of saying that. It is determined by the attractions and repulsions between the side groups, or those R groups of the amino acids. So that's important. I want you to know that tertiary structure is held together by those side groups. So here are some interactions holding the tertiary structure together. So what are some interactions between those R groups? They call that cross-linking. So cross-linking in your tertiary structure involves the attraction and repulsion between those side chains. I've already mentioned that, and here are some of them. So hydrophobic. Before you even go on, without reading this, what amino acids, what was our classification of amino acids that were hydrophobic? So you remember you had those 4 classifications. They were non-polar amino acids, polar, acidic, and basic. So if I'm asking you what cross-linking do you think is happening between that are hydrophobic interactions, I hope you say that it's between those non-polar amino acids. Those amino acids that the R group was just hydrocarbons. So it says the hydrophobic interactions are the attractions between your non-polar groups, those non-polar amino acids. Hydrophilic interactions are going to be between your polar groups and water. And why they have the water in there is because our proteins, our bodies are mainly made up of water. And so the way the protein is going to fold has a lot to do with the R groups being either attracted to another R group or being attracted to the water that is surrounding this protein. Salt bridges, if you remember from our acid-base chapter, and we talked about it, an acid and a base, the neutralization reaction, it forms a salt and water. That's one of the products of a neutralization reaction. So when you have an acid and a base reacting, they often form what we call a salt. So that's where the term salt bridge comes from. So a salt bridge cross-linking is that ionic interaction, because it's a cation to an anion, between an acidic amino acid and a basic amino acid. Hydrogen bonding is going to occur between when you have a hydrogen directly bonded to an oxygen in your amino acid R group, or a hydrogen directly bonded to a nitrogen. And then the last one is disulfide bond, and that's only going to occur once, and that's between 2 cysteine molecules, because cysteine is the only thiol we had. So it is the strong covalent link between sulfur atoms when you have two cysteine amino acids bond. So I do want you to know these. Okay, so there's 5 cross-linkings that occur, and I want you to be able to point them out or know which one they are. Okay, here's a picture kind of showing us those different cross-links. What they do in biochemistry, these proteins are so huge and the molecules have so many atoms in them that what they do to try to simplify it is they do what we call this ribbon structure, where they take the backbone of the polypeptide or protein, and that's what they symbolize as this little blue ribbon. Because what you're mainly focusing on then is the R groups, because it's the R groups that determine that three-dimensional tertiary structure. So looking at the ribbon, see if you can identify the alpha helix and the beta sheet. And hopefully you said right here is the alpha helix, and right here with the hairpin curve, they're telling us it is our beta sheet. Okay, so we have our helix. And if I were to ask you what these little red dots, what type of a bond this was, I would hope you would say it's the hydrogen bonds that are occurring along the backbone of the primary structure. It's not hydrogen bonding that's happening between the R groups. Okay, so those are our secondary structure. Focusing on the interactions, the different cross-linkings that's happening, if we were to focus on this one right here, what would you say? And I hope you would see that you have this OH group, and the hydrogen is interacting with either an oxygen or a nitrogen, so you would say this one here is a hydrogen bonding cross-link. Same thing with this one. Here's the OH group, and here's the NH, so this would also be a hydrogen bond cross-link. Down here, what kind of an interaction do we have here? We've got 2 different amino acids hanging out together, this guy and this one, and they're both just hydrocarbons. So I would hope you would say this one right here is a hydrophobic interaction. If we were to look at this one right here, this is our disulfide. So this would be our disulfide cross-link, because we see 2 sulfurs bonded together. Right here, a big giveaway is this positive and negative sign, because that would be our cation to our anion, which is an ionic interaction, which is what we call the salt. So this would be the salt bridge. It also can be called just an ionic interaction, or an ionic cross-link. Okay, that looks like we've got all those. So here's a learning check. For the sake of this video, you have all of these amino acids on the next slide. So to answer this slide, you have to go back and forth. But for the taping purposes, I went and I cut and pasted the particular amino acids we needed to answer this. So select the type of tertiary interaction that would cause a disulfide bond, hydrogen bonds, ionic, and hydrophobic. So if you put leucine, if the R group from leucine is interacting with the R group from valine, what kind of a cross-link would that be? So if I look at valine and I look at its R group here, it's just a hydrocarbon. If I look at leucine, it is just a hydrocarbon. So what kind of an interaction is going to happen between these 2 R groups? And I'd like you to say hydrophobic. So this interaction would be hydrophobic, number 4. If you have 2 cysteine amino acids, what kind of an interaction is that going to be? Well, here is my thiol. Remember, if you have 2 thiols, that's going to make that disulfide bond. So the answer to B is going to be number 1, the disulfide. Is 4. C is aspartic acid with lysine. So when we look at the R group of acetic acid, that is our carboxylic acid. And lysine is a base, so that's going to give us the ionic interaction. Or another word for the ionic interaction would be a salt bridge cross-link. Serine and threonine, I have an OH group interacting with another OH group, so that is going to be hydrogen bonding. So that would be a number 3. This is a picture, the tertiary structure of the enzyme carboxypeptidase. And I like the picture. It's a really, they're not showing you all the atoms. It's kind of a simpler model. And I like it because it gives you an idea of how huge these molecules are. This is yeah, enzymes and proteins are just really big molecules. So they get complicated, and it takes computers a long time to, to optimize these structures and come up with the shapes of them. Carboxypeptidase, I think you'll learn more about it in biology, but it's the enzyme that actually will hydrolyze the peptide bond at the carboxylic end of the primary structure of a molecule. I'm not going to ask you any questions about that. I just kind of wanted you to get a feel for how big these guys are. Okay, so the quaternary structure. It is the combination of two or more protein units. So sometimes you'll have one protein that has collapsed into its tertiary structure and another protein that has collapsed into a tertiary structure, and then those two or more proteins will hang out together, and they'll also be attracted to each other by the same R group interactions that hold the tertiary structure together. So, and the giveaway that they're talking about the quaternary structure is the two or more protein units. It's almost always going to say that in the definition of the quaternary structure. The quaternary structure of hemoglobin consists of 4 peptide chains as different subunits. So here's a picture of hemoglobin. Here is the heme holding the iron inside each one. But if you notice, we have 4 different proteins that are hanging out together. So that would give us the quaternary structure. The quaternary structure is stabilized by the same interactions found in the tertiary structures. So it would still have all 4 of those same cross-linkings, or 5 cross-linkings, that hold the tertiary structure together. So once again, it's the R groups that are holding the quaternary structure together. So here's a summary of the protein structures. The primary is the sequence of amino acids. In the secondary, I want you to know the two, the alpha helix and the beta sheet. So there's the alpha helix, beta pleated sheet. We're not getting into the triple helix in this class, but I do want you to know they're held together by hydrogen bonding along the chain, the backbone of the chain. Your tertiary structure is the folding of the protein into a compact 3-dimensional shape, stabilized by interactions of the R groups. And then the quaternary structure is a combination of 2 or more proteins. Okay, so that's kind of a summary of what I want you to know about protein structure. Here's a picture that kind of shows it. I kind of like it. There's our primary structure, just the sequence. Here they're showing us an alpha helix to represent the secondary structure. The tertiary structure is the folding of the protein upon itself, and then there's a picture of the quaternary structure. So a quick learning check. Identify the level of protein structure as either primary, secondary, tertiary, or quaternary. A is a beta pleated sheet. So that would definitely be secondary, which is our number 2. The order of amino acids. So that is our primary structure. The primary structure just gives you the order the amino acids are bonded to each other. A protein with 2 or more peptide chains, that is going to be the quaternary. The shape of a globular protein, this one's a little bit tricky. I didn't talk about this, but when you have your primary structure, let's say, and then parts of it have folded into some secondary structure. So I've got this big long primary structure with some alpha helix and maybe some beta sheets. But if I put that in water, it is going to, all of the R groups that are hydrophobic are not going to want to be around the water. So they're going to all clump together and try to get away from the water. And what happens is it causes the protein to fold into this shape where all the inside of the protein has all the hydrophobic area, and all the hydrophilic R groups that want to be around the water tend to be on the outside. So often when you put a polypeptide or a protein into a water environment, it's going to make a globular protein. So kind of almost a spherical shape with all the hydrophobic areas on the inside. So a globular protein would be an example of the tertiary structure. That one was a little bit tricky. Oh, excuse me. E is the disulfide bonds between the R groups. That one can be either tertiary or the quaternary, because I told you it's the same interactions. Usually we just talk about that in the tertiary structure. But for the sake of this class, I told you that the same types of cross-linking that happens in tertiary structures is what holds the quaternary structures together. Okay, denaturation. It involves the disruption of bonds in the secondary, tertiary, and quaternary structures. It does not break apart the peptide bonds in the primary structure. That's important to know. So here's some ways to denature a protein. Heat and organic compounds will break apart the hydrogen bonds and disrupt any hydrophobic interaction. So organic compounds tend to be nonpolar for the most part. So if you put a protein into an organic solvent that's nonpolar, well, originally all the hydrophobic R groups were in the center. But now if you put them in an environment that's more nonpolar, it's gonna totally make the protein denature and fold into a different type of shape and not function the way it's supposed to. You can use acids and bases will denature a protein. It's going to break apart the hydrogen bonds between the polar R group and disrupt any ionic bonds or salt bridges. Heavy metals, some of them will actually break the disulfide bonds. Agitation, so just basically like whipping your egg. So if you can agitate it really hard, you can actually break those interactions and denature the protein. It kind of stretches out the peptide bond. So I do want you to know these different ways. You don't need to know what bonds are interrupted, what cross-linking is interrupted by these, but I would like you to know the different ways to denature a protein. What else? Yeah. Here's a picture, so an application of denaturing a protein. We see that when we cook an egg. So you have your egg and then you heat it up, and the heat is going to cause the protein, all those secondary, tertiary, and quaternary interactions to break. So notice that the primary structure does not break. That ribbon stays in contact. So when you cook an egg, you denature proteins. When you wipe your skin with an alcohol wipe, that's going to denature some proteins, especially in the bacteria and things like that you'd have on your skin. When you use heat to cauterize blood vessels, instruments are sterilized in an autoclave, and that's going to denature proteins. So, all right, the favorite slide, the end. You guys did it. This is a hard class, and so you should be really proud of yourself you made it to this point. Congratulations.